Today I'm going to be talking to you about the different structures that make up a DNA strand. This here is my sample DNA strand, and first we're going to be talking about what makes up a nucleotide, which is what makes up a DNA strand. So first, on the DNA strand, we have the little black beads representing a phosphate. Then the big white beads are going to be deoxyribose sugar. The blue rectangle is going to be thymine. The yellow rectangle is going to be adenine, and the red rectangle is going to be guanine. The white rectangle is cytosine. Base pairing is what happens between the nitrogenous bases. So adenine pairs up with thymine in DNA and uracil in RNA. Then, as well, guanine pairs up with cytosine. Let's take a dive deeper into what exactly these nitrogenous bases are. So again, here are the base pairings. Adenine and thymine go together, and cytosine and guanine go together. I remember them because adenine and thymine form a word, at, and then the other one, cytosine and guanine, their first letters have a similar shape. As you can see, adenine and thymine have 2 hydrogen bonds, whereas cytosine and guanine have 3 hydrogen bonds. A way to classify these bases is purines and pyrimidines. Adenine and guanine are purines with 2 rings, and cytosine, thymine, and uracil and RNA are pyrimidines with 1 ring. A way that I remember adenine and guanine as purines is that ag is appear and honest work, and then cytosine and thymine, or CT, is prime for a diagnosis. Coming back to our DNA model now, we're going to talk about the different bonds that hold between the nitrogenous bases. So between these nitrogenous bases here are hydrogen bonds that keep them together. There are also hydrogen bonds between them, like so, that help stabilize the DNA. If you were looking at the phosphate backbone of the DNA, that would be having covalent bonds, which are more sturdier. Let's take a more in-detailed look about the phosphate backbone. A sugar has 5 carbons, which are denoted as primes, and the phosphate group binds to the 5' end of the one sugar and to the 3' end of the other sugar. The 1' end is where the nitrogenous base adds to the sugar. Another thing I want to mention is the handedness of DNA, which is basically the direction in which the DNA is going or curving. I figured this out by drawing out an upward arrow along the curve of the DNA, so for this one it would be left-handedness. Also, DNA strands are anti-parallel, meaning they run opposite of each other. So as you can see here, on one side it has a 3' end, and the other side is a 5' end.